When we duplicate DNA, our goal is to start with 2 template strands of DNA and then add a complementary strand to each of these old strands to create 2 new double-stranded pieces of DNA. The first step in doing that is to unwind the helix so that we can gain access to all of the nucleotides that are inside of the DNA double helix. So the DNA is going to start out in this double helix structure where our backbone is going to be crisscrossing around and swirled at the same time. We've got our bases joined with hydrogen bonds in the middle. But since they're bound to each other, we can't gain access to them unless we unwind them. So the very first step is going to be to turn the double helix into a flatter, like 2-dimensional almost structure, and then to open it up so that way we can see what is inside, kind of like as though you're going to open up the cover of a book so that you can start to read whatever is inside of that book. And once we've opened it up, the bases will be on the inside. Let's just throw in some bases. Let's say it's like G, C, C, A, and then C, G, G, T, the complement to the template. The way that we go from this double helix structure to this opened structure is with 2 enzymes that you're going to find outside of this replication bubble that are called Helicase and Topo Isomerase. When I draw them, you notice I got like one on the left and one on the right, but left and right isn't a good way to think about helicase and topoisomerase. Instead, think about inside and outside. Helicase is on the inside. Helicase can be found there and there, and it is moving outwards from the replication bubble, unzipping the double helix as it goes. So if you got a zipper and you got the pull that you're moving up and down, the pull that you're moving up and down would be like helicase, and it's unwinding the DNA. The problem is that the DNA is wrapped around itself, and as you grab it in the middle to unwind it, imagine you got a piece of string and you're grabbing the string in the middle to unwind it, you're going to put tension on the parts of the string that have not been opened over here on the sides, because every time that you unwind the center, you're winding these edges tighter and tighter and tighter. So we have to relieve that tension, otherwise we're going to break the DNA. And the enzyme that is on the outside of the bubble that is relieving that tension is called Topo Isomerase. And the way that Topo Isomerase is going to relieve that tension is it cuts the DNA, allows the DNA to spin out to get rid of that tension, and then it'll glue it back together again. And so Topo Isomerase is on the outside, helicase is towards the center. Helicase is unwinding the DNA, Topo Isomerase is cutting the DNA, letting it relieve the tension, and then gluing it back together again to prevent the DNA from breaking itself as it opens from the double helix. As soon as we got it open, the other thing that's going to happen is we're going to have to bind something to keep it from just snapping right back to the way that it already was. And so if we just kind of duplicate what we have over there, let's put some bases in. I'm going to go with black. Put some bases inside of here. So let's say this is G, C, C, A, T, G. This would be the complementary strand, so it'd be C, G, G, T, A, C. As soon as we open up this helix, again, it's going to want it to snap back together because the C is attracted to the G, the T is attracted to the A. These guys just want to go back the way they were. That's why they were like that. So we have to cap them. We've got to put something on top of each of these bases to prevent them from just snapping back. And so we're going to put little bitty proteins, there's going to be a whole bunch of them, onto each one of these bases. And they'd be going all the way across. For right now, I'm only going to draw some over here on the right. But these proteins that we're adding, just to keep it so that it's happy and it doesn't immediately snap back, are called single-stranded binding proteins. For short, they're called SSBPs. And their job is just to bind to the bases to keep the helix open after it's been opened by helicase and topoisomerase. So if I was to draw helicase and topoisomerase again, they'd be here and there. Helicase is towards the inside, topoisomerase is towards the outside. Topoisomerase is always towards the outside, again, because that's where the tension is building. you're not going to have tension towards the inside. The tension's going to be outside of the bubble. And so that's why Topo Isomerase is always on the outside. Over here where you got the bases open, we want to start making our other complement of DNA. But the enzyme that makes DNA, DNA polymerase, can't attach directly to the strand of DNA and just start spitting new molecules of DNA out. It needs to have some kind of little starter segment here, some kind of little miniature starter piece that is attached to the template so that way the DNA polymerase has somewhere to attach when it comes in to do its work. So we're going to have another enzyme come in, and this enzyme is called primase. Primase is going to swoop in to the beginning right here of this open area, and it's going to read the DNA from the 3' to the 5' direction. So in my drawing, this side would be 3', this side would be 5', and it's going to go along that template. In this instance, it'd be from left to right because 3' on the left. So as primase moves from here to there, it's going to make a primer. A primer is a short segment of DNA. So I'm just going to have it be 2 bases long here. That's probably unrealistically short, but it'll just be a little piece. And it's complementary, so this would be a C, this would be a G. The difference is that your primer is made from RNA. It's just a single strand, it's temporary, it's not as long-lived. And primase will make this primer so that way in the next step, DNA polymerase can attach to that little segment of RNA and start laying down actual DNA that is the complement of that template. So in the next step, primase is going to leave. It's going to come in, make the primer, and then leave. And then the next enzyme, DNA polymerase, is going to be able to attach. So just got to give myself my bases again. And your primer doesn't leave, so my primer would still be here at this position. A C and a G, and again, that's my primer. But now a new enzyme, DNA polymerase, is going to come in. It's going to attach to this location where you got the primer bound to the template, and it's going to start moving from the 3' to the 5' direction just like the primer did. The difference is that DNA polymerase makes DNA. So as DNA polymerase moves, and this picture would be from the left to the right, it will be laying down DNA instead of RNA. And so the DNA is going to have a G and a T and then an A. And it's just going to keep on going until it hits some obstacle or comes to the end of the entire piece of DNA.